Hi students, this is a recording to help you complete the Microscope Lab. And so first what I'm going to do is I'm going to show you how to get to the Microscope Lab. So this is the course home page. To get to the Microscope Lab, you can go to Unit 1, the Science of Biology, then scroll down until you get to 1.10, Laboratory Using a Microscope. That'll give you all of the options here. So basically, you can go ahead and go through each of these slides to read on different things about how to handle a microscope and how to use a microscope. So you're going to go ahead and read through these slides here. And then it'll take you to slides that show you how to handle a microscope, carry a microscope, and store a microscope properly, how to keep the microscope clean, and the different parts of the microscope. Okay, so you should definitely read through these before completing the lab. Afterwards, it's going to take you to Investigation Using a Microscope. You want to go to page 2 of 2, and at the bottom here it says Using a Microscope Virtual Lab. So you click that and it'll take you to another link where it says that you are leaving your course content. You want to click the link below and it'll take you to the virtual lab. Okay, so it's going to be helpful for you to view the tutorial as well, but today I'm not going to go over the tutorial. I'm just going to go ahead and help you begin the lab. Okay, so you'll press Begin the Lab. While you're doing that lab though, I want you guys to also open up the document that you'll need in order to complete the lab. So you can press the next arrow, and you should see Digital Materials Lab 1.10 Using a Microscope. So you'll click that and you'll go ahead and open the file. Okay. You want to make sure to put your name as well as the date, and then you'll go ahead and go through these questions and answer them for the lab report. Okay, so I'll go ahead and walk you through 1 through 3, and then we'll go ahead and do the lab, and I'll walk you through questions 4 and 5. So for question 1, how do you calculate magnification on a microscope? So in order to calculate magnification in a microscope, you need to multiply two things: the magnification of the ocular lens and the magnification of the objective lens, which is 4x, 10x, and 40x. Okay, so those are some examples of magnifications on an objective lens. It's important that you know that even though you're using a 4x or 10x or 40x objective lens, that doesn't mean that you're magnifying to those amounts. You always need to multiply by the magnification of the ocular lens to get the total magnification. The second question that you need to answer is explain how to focus a microscope using the high power lens. Include any safety issues you need to be aware of. So you're answering two questions: how do you focus a microscope using a high power lens? And then you also want to mention any safety issues that you might need to be aware of. Okay, so in order to focus using a high power lens, you do need to use a part of a microscope called the focus adjustment knobs, and you can see it right here. So you're going to turn this knob to focus your image, and how it focuses your image is that it actually moves this stage portion right here up and down. And so it's going to move it closer to the lens or further from the lens so that you can focus your image. There are two different types of focus adjustment knobs. We have the fine adjustment knob and the coarse adjustment knob. And we'll go over the difference of the two. Remember, in this question, it also asked you, what are the safety concerns when you are focusing an image? Remember that because these knobs move the stage up and down, sometimes you can move the stage too far up where it actually meets the objective lens, which can damage the slide and damage the microscope. So it's really important that you don't turn the knob too much that the stage collides with the objective lens. Okay, so we're going to go over now the difference between the two different focus adjustment knobs and how you use them to focus your image. Okay, so this is a close-up of looking at the letter E. Okay, so when you put the slide onto the microscope, you're going to see that you don't really see much at all, right? You might see a little bit of gray color showing you that there's probably something there, but you can't actually see the image. So what you're first going to do is you're going to use the coarse adjustment knob. And the coarse adjustment knob moves the stage up and down for focusing, and it moves it quite a bit. And so it helps you generally pick up or generally position the stage where you can actually see the object. But we can see here that the edges aren't very crisp. So we want to use the fine adjustment knob like you see here, and we want to turn it until we actually get crisp edges and a sharp image. And the fine adjustment knob does this by moving the stage slightly up and down, very, very slowly and not at great amounts. Whereas the coarse adjustment knob has more drastic movements of the stage. And so again, when you want to focus an image using a high power lens, you want to first use the coarse adjustment knob and then use the fine adjustment knob. Okay, so the next question is, why is it important to keep a microscope covered when you're not using it? And so a microscope actually has a cover that covers the entire thing, and it's important to have it covered because there's things like dust that can fall into the microscope and make it dirty. And obviously you wouldn't want to use a dirty microscope because you wouldn't be able to see through the eyepiece and see the slide properly as well. And so it's really important that you keep each of the microscopes covered so that it's protected from dust, so that when you use it, you can see everything very clearly. All right, so those are the first 3 questions in the lab report. You should be able to answer those now. The next 2 questions is going to require us to actually go into the lab where we're going to be observing the onion root tip. And we're also going to be observing the brine shrimp in the hay infusion. Okay? For each of these, we're going to either describe in words or sketch what we observe using the low power lens and the high power lens. So the low power lens is what we're going to call 10x, and the high power lens is what we're going to call 40x. So essentially what we need to do is we need to look at each of these things under the microscope using the 10x and the 40x and either sketch or describe in words what we see when we use these 2 different types of lenses. So let's go ahead and go into the virtual lab. So we had the virtual lab open from before, and so you want to first do the tutorial. Once you're done with that, then you can click Begin the Lab. And you can pick any glove color, doesn't matter. And then press Start. Once you do that, you'll see that the microscope is covered to protect it from dust, so you're going to want to take that cover off and put it to the side. Then you're going to want to prepare the microscope so that we can use it. The first thing is we need to clean the microscope with the lens paper. Make sure you're clicking it so that it cleans that eyepiece. And then we can go ahead and throw it away in the trash. It's going to ask you if you want to do this because it's permanent and you want to press yes. After we have the microscope all clean, we need to turn the microscope on and turn the light all the way so that it's at its full brightness. The next thing is that you want to swivel this nose piece right here, and we want to start at 4x, which is the lowest magnification, and we call this the scanning lens so that we can find the object that we're trying to look at. Remember, the first thing we were trying to look at is the onion root, so we can go ahead and pick up the onion root and put it under the microscope. You can actually move the stage around like this and try to find the onion root. You can see that there's some sort of pinkish or reddish image here. Obviously we can't see much, but we know that we are looking at something. So what we're going to do now is look at it under the low lens, which is the 10x. So we have to swivel it one more time. So you should see 10x, which should be the yellow lens. Remember, when we're trying to focus using the low or high power lens, you need to first use the coarse adjustment knob, which is behind here. You're going to turn it until you see a pretty sharp image. Okay. To make it a little bit sharper, you can try moving the fine adjustment knob, which is in the front, to make a sharper image. Just something like that. Okay, so this is what the onion root tip looks like under the 10x or low power lens. So you want to describe it in words, or you can sketch it. Or if you know how to take a screenshot, you can also put it a picture of it as well. Okay, the next thing we need to do is look at it at a low power lens. So you can go ahead and swivel the objective lens, and you should be at the blue lens, which is the 40x. So obviously now it's out of focus again, so we need to adjust the coarse adjustment knob. Do you see how it asks, do you want to keep turning the knob? You want to say no, and the reason why is because it's telling you if you keep turning the knob, it's eventually going to break the slide. So I'm going to turn it the other way, try to focus it, and then I'm going to focus it a little bit more using the fine adjustment knob. And I don't really see much here, so I'm going to try moving it around to see if I can see something a little bit more interesting. Yeah, so maybe I can describe something like what this looks like here. Okay, so then you want to sketch or describe what you see using the 40x lens or the high power lens in your lab. All right. So after you do the onion root, you can go ahead and swivel it back to 4x where we use the scanning lens, and we're going to take off, we're going to take off the slide and place it back where we had it. The next thing we wanted to look at was the hay infusion that has the brine shrimp in it. Okay, so how you're going to do this is actually a wet sample, so we have to prepare it. It can't be prepared for us. So we'll take a dropper, get some of the hay infusion, and put it on the slide. Then we can put the dropper back down. When you prepare a wet slide, you actually need a cover slip to cover the wet sample. And so you'll take the tweezers or the forceps, take a cover slip, and place it on the slide. Then you can put the forceps down. Now you have your slide prepared, so you can then bring it underneath the microscope. So I'm going to move it around to make sure we see something. Sorry, I'm having a hard time adjusting it. Okay, I think there's something right there. So then you want to go ahead and swivel it to 10x. Let me try to see if I can find the object. There we go. Okay, and then you'll go ahead and use the coarse adjustment knob to focus on it, and then the fine adjustment knob to sharpen the image. Okay, and then you can go ahead and draw that, or you can describe in words. Then you can go ahead and swivel it one more time to 40x, where you can sharpen the image with the coarse adjustment knob, and then use the fine one as well. Okay, and you want to sketch and describe that in words. Right. Again, remember that you need to answer it for the low power lens, which is the 10x, and the high power lens, which is the 40x, for both of the onion root tip as well as the brine shrimp in the hay infusion. After you're done with this, you're going to then submit it into Dropbox. And so how you submit it to Dropbox is you're going to go back to the course page, you go to Tools, go to Dropbox, and then you can press Using a Microscope 1.10 Lab, and you can upload it there. So just make sure you're saving the lab to your computer so that you're able to upload it. If you have any other questions, you can email me or come to a help session on Tuesday or Thursday at 12:30 to 1 p.m.